All right, let's dive in. Today, we're going way back to the basics.
Back to the building blocks, you could say.
Exactly, the fascinating history of DNA and RNA.
How we went from not even knowing they existed to, well, everything we know now.
It's amazing what we've learned, right? And for this deep dive, we've got some incredible material to work with.
Oh yeah, some real gems.
Like Microbiology, Q&A 9, Biochemistry of the Genome.
A treasure trove of insights in that one.
So we're not just going to lay out facts, we're going to trace the steps, the detective work.
The how and the why, those aha moments.
Right, because these molecules, they might seem simple, but they hold the blueprint for life itself.
It's mind-blowing when you think about it.
Absolutely. So let's start with this. DNA was actually discovered long before anyone knew what it did.
Way before we knew it was the molecule of heredity.
Like way back in 1869.
A Swiss chemist, Friedrich Miescher,
Studying white blood cells, of all things.
found this phosphorus-rich compound he called nuclein.
And that, my friends, is what we now know as DNA.
Classic case of scientific serendipity.
Right. He wasn't even looking for it. Now, around the same time,
We have Gregor Mendel meticulously crossbreeding his pea plants.
the 1850s and 60s, tracking traits, flower color, seed shape.
This is all before anyone even knew DNA existed.
But his work, it laid the groundwork for understanding how traits are passed down.
The principles of inheritance, even without knowing the mechanism behind it.
So on one hand, we have this mysterious nuclein, and on the other, the laws of inheritance.
But the connection, the missing link, that took a bit longer.
Fast forward a few decades to the 1930s and 40s, we meet Joachim Hemmerling.
And his fascinating work with a giant alga.
Acetabularia, a single-celled organism, but you can see it with the naked eye.
It's remarkable, really.
And Hemmerling was all about the nucleus, the control center.
He'd graft different parts of these alga cells together.
And what did he find?
That the nucleus, it called the shots, even in a single-celled organism.
It controlled development, like a tiny conductor leading the orchestra.
This was huge, linking the nucleus directly to heredity.
Okay, so the nucleus is in charge, but how does it actually exert its control?
Well, that's where we enter the world of bread mold.
Bread mold.
Sounds unassuming, but it's scientifically invaluable. Notospora crassa, to be exact.
This is where Beadle and Tatum come in, 1940s.
They were looking at mutations in the mold, ones that messed up specific metabolic pathways.
Metabolic pathways, those chains of chemical reactions in a cell.
Right, and by analyzing these mutations, they came up with a groundbreaking idea.
The one gene, one enzyme hypothesis.
Each gene, they proposed, holds the instructions for making a specific enzyme.
Enzymes, those proteins that speed up chemical reactions.
This was a direct link between genes and proteins, a huge leap forward.
But still, we didn't know what molecule was actually carrying the genetic information.
So the mystery continued, but we were getting closer.
Now, this is where things take a turn. We enter the world of bacteria.
Bacterial transformation, where the evidence for DNA as the hereditary molecule starts piling up.
Let's talk about Frederick Griffith, 1928, and his work with Streptococcus pneumonia.
Two forms of this bacteria, a harmless rough strain and a deadly smooth strain.
He was trying to figure out what made the smooth strain so virulent.
His experiments led to a startling observation.
He injected mice with a mix of heat-killed smooth bacteria and live rough bacteria.
Now, the heat-killed smooth ones should have been harmless.
But the mice, they died.
And what's more, he found live smooth bacteria in their blood.
So something from those dead smooth bacteria, it transformed the harmless ones.
We call this phenomenon transformation.
It's like something out of a sci-fi movie.
Griffith couldn't pin down what caused it, but it was a crucial step.
Took another 16 years before Avery, McLeod, and McCarty picked up the trail.
They systematically destroyed different parts of those heat-killed smooth bacteria.
Proteins, lipids, carbohydrates, DNA, one by one.
And they tested if the remaining mixture could still transform the rough bacteria.
They were like molecular detectives isolating the transforming principle.
And their results were groundbreaking.
When they destroyed DNA, the transformation stopped.
It was the smoking gun. DNA was the transforming principle.
The molecule responsible for carrying the genetic instructions.
It was a huge leap forward.
Yeah.
But some scientists remained unconvinced.
Of course, this was about identifying the very molecule of inheritance. It had to be airtight.
So enter Hershey and Chase, 1952, with their elegant experiment using viruses.
Bacteriophages, those viruses that infect bacteria.
They knew these phages inject their genetic material into bacteria, but was it protein or DNA?
So they devised a brilliant plan to tag these molecules.
Radioactive labels, sulfur for protein, phosphorus for DNA.
Then they let the labeled viruses infect bacteria.
And get this, they used a blender to separate the viral parts from the infected bacteria.
A blender in a genetics lab.
It did the trick, shearing off the viral coat.
And the results were clear.
The sulfur, tagging the protein, stayed mostly with the viral coats.
But the phosphorus tagging the DNA, it was inside the bacteria?
Definitive proof. It was DNA carrying the instructions to make more viruses.
The Hershey-Chase experiment, a game changer.
It settled the debate. DNA was the molecule of heredity.
So from mysterious nuclein to deadly bacteria and virus-wielding blenders, what a journey.
It highlights the dedication and creativity of these scientific pioneers.
Absolutely. Their work laid the foundation for everything we know about genetics today. All right, so last time, left off with that incredible Hershey-Chase experiment.
Really solidified DNA as the carrier of genetic information.
But knowing DNA is the key is one thing. Understanding how it works, that's another.
Right, it's like having a treasure chest, but not knowing how to open it.
So let's unlock that chest, shall we? Let's zoom in on the structure of DNA itself.
The structure is truly elegant, and it reveals a lot about how this molecule functions.
You always hear it described as a double helix, right?
Exactly, like a twisted ladder.
Okay, I can picture that. So what forms the sides of this ladder?
That would be the sugar-phosphate backbone.
Backbone, so it's the structural framework.
You got each strand of DNA has this backbone, alternating sugar and phosphate molecules.
Strong bonds holding it all together.
Oh yeah, those are called phosphodiester bonds.
Okay, so that's the sides. What about the rungs connecting the sides?
That's where the nitrogenous bases come in.
Adenine, thymine, cytosine, guanine, right?
Yes, A, T, C, and G, the 4 bases of DNA.
And they pair up in a very specific way. A with T, C with G.
Always. That's complementary base pairing.
Like puzzle pieces fitting together.
The perfect analogy, and it's crucial, because it means the 2 strands are complementary.
Like 2 sides of the same coin.
Exactly. They essentially hold the same information.
This base pairing, how did scientists figure that out?
Well, it was a combination of clever analysis and some high-tech imaging.
Okay, tell me more.
Erwin Chargaff, late 1940s, he analyzed the base composition of DNA from different organisms.
Well, he was looking at the amounts of each base.
And he found a striking pattern. The amount of adenine always equaled the amount of thymine.
Same for cytosine and guanine.
Exactly. That was the first big clue that they were pairing up.
Like a 1-to-1 ratio.
Precisely. Then we have Rosalind Franklin and Maurice Wilkins using X-ray crystallography.
Now that sounds complicated.
It's a technique using X-rays to figure out the 3D structure of molecules.
So they were essentially taking pictures of DNA.
In a way, yes. And these images revealed the helical structure of DNA, that double helix shape.
Then they showed it had a consistent diameter.
Right, all thanks to those X-ray pictures.
So those images, combined with Chargaff's rules about base pairing, led to the big breakthrough.
Ah, yes. The famous double helix model by Watson and Crick in 1953.
Truly iconic, right? This elegant structure holding the blueprint for life.
It's a testament to the power of collaboration and, well, the beauty of nature's design.
Okay, so we have this double helix, the backbone, the base pairs forming the rungs.
But there's a twist, literally.
What do you mean?
The 2 strands, they don't run side by side, they run in opposite directions.
Oh, I remember this anti-parallel orientation, right?
You got it. One strand goes 5 to 3, the other 3 to 5.
You know those numbers, they refer to the carbon atoms in the sugar molecule?
That's right. And this orientation, it's crucial for things like DNA replication.
Okay, so that's DNA, but what about its close cousin, RNA?
RNA, it's similar, but with some key differences.
Like what?
First off, RNA is usually single-stranded.
So like 1/2 of the DNA ladder.
You could say that second, RNA uses a different sugar, ribose, instead of deoxyribose.
Okay, so different sugar, and there was another difference, right?
Yes, RNA uses uracil, U, instead of thymine, T, as one of its bases.
So different sugar, different base, single-stranded. Why is RNA so important then?
Well, it plays a vital role in translating that DNA information into proteins.
It's like the middleman.
Precisely, carrying the genetic code from the nucleus to the ribosomes where proteins are made.
This is what we call the central dogma of molecular biology, right?
Okay.
DNA to RNA to protein.
Exactly, and there are different types of RNA involved in this process.
I remember learning about messenger RNA, mRNA. It carries a code.
Like a messenger carrying instructions, yes.
Then there's ribosomal RNA, rRNA, part of the ribosomes themselves.
Those protein-making factories, and rRNA is part of the machinery.
And finally, transfer RNA, tRNA, it brings the amino acids to the ribosomes.
Amino acids, the building blocks of proteins, and tRNA has this unique structure.
It recognizes both the code on the mRNA and the corresponding amino acid.
Like a tiny molecular delivery truck, bringing the right part to the right place.
This whole process, it's mind-boggling.
It is a perfectly coordinated molecular dance.
The central dogma, it really underpins all of life.
It does, from the simplest bacteria to us.
Okay, so we've gone from DNA's double helix to the different types of RNA.
All working together to build proteins.
Now let's zoom out a bit and talk about genomes.
The complete set of DNA for an organism, all its genetic instructions.
Like the instruction manual for building and maintaining life.
You got it. And genomes can be surprisingly complex, even in simple organisms.
Prokaryotes, like bacteria, they have a single circular chromosome.
And eukaryotes, like us, multiple linear chromosomes, all neatly packaged in the nucleus.
But here's something I always found fascinating. A lot of our DNA doesn't code for proteins.
Ah, yes, the non-coding DNA used to be called junk DNA.
But we're learning it's not junk at all.
Not at all. It plays many roles, regulating gene expression, organizing chromosomes.
Like switches and scaffolding.
And some of it might be remnants of ancient viral infections or evolutionary leftovers.
So the genome, it's this vast, complex landscape, still holding many secrets.
Absolutely. And that's what makes genetics so exciting, always something new to discover.
Well, from the double helix of DNA to the roles of RNA and the vastness of the genome.
We've covered a lot of ground tracing the flow of genetic information.
But the journey doesn't end here. In our final part, we'll explore the implications of all this.
The impact on understanding life, health, and the future of genetics itself.
Okay, so we've delved into this microscopic world, DNA's structure, RNA's role, the genome.
We've seen how science has uncovered all this, but the big question is, what does it all mean?
Right. What are the implications for us, for understanding life itself, health, disease, the whole picture?
Well, these discoveries about DNA and RNA, they've completely revolutionized biology, medicine, everything.
It's not just about molecules anymore. It's about what they tell us about life on Earth.
Exactly. It's given us these powerful tools to understand life's basis, develop new treatments. It's incredible.
Let's start with the big picture, understanding life itself. How has all this changed our view of evolution?
Well, one of the most profound things we've learned is the universality of the genetic code.
That every living thing uses the same basic code.
It points to this common ancestor, this shared origin for all life on Earth.
So we're all connected by this language of DNA and RNA.
Precisely. And by comparing genomes, we can trace those relationships, see how life branched out over millions of years.
It's like having a molecular family tree.
And it's not just about the past, it's about the future too, the future of medicine.
Right. This is where things get really exciting, or maybe a bit daunting too.
We can diagnose genetic disorders now, screen for susceptibility, even tailor treatments based on your genes.
This is what they call personalized medicine. Incredible, but it raises a lot of questions too.
Oh, absolutely. Ethical considerations are huge here, especially when we talk about gene editing.
Gene editing, being able to actually change the code, that's powerful stuff.
Powerful and potentially transformative CRISPR-Cas9, these technologies let us alter DNA sequences with precision.
Like something out of science fiction, treating or even curing genetic diseases.
The possibilities are incredible, but we have to proceed with caution.
We need those open discussions about the ethics, ensure these tools are used responsibly.
It's a delicate balance, the power to change the very essence of life.
So DNA and RNA, they haven't just changed our understanding, they've challenged us, pushed us to think about the future.
And these questions will continue to evolve as we learn more about the genome, about the potential of these technologies.
Well, I think that about wraps up our deep dive. From those early discoveries to the complex dance of molecules.
From nuclein to genomes, we've covered a lot of ground.
And I hope everyone listening has gained a new appreciation for these incredible molecules.
For their elegance, their complexity, their central role in all of life.
It's been a fascinating journey, and as always, the exploration continues.
There's so much more to learn, so keep those minds curious, folks. Never stop questioning, never stop exploring.
Because the world of science, it's full of wonders just waiting to be discovered. Thanks for joining us.